Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Environmental contaminants like fluoride, lead and pesticides exist all around and even within us. While researchers have simple ways to measure concentrations of such contaminants inside lab environments, levels are much more difficult to test in the field. That's because they require costly specialized equipment. Recent efforts in synthetic biology have leveraged cellular biosensors to both detect and report environmental contaminants in a cost-effective and field-deployable manner. Even as progress is being made, scientists have struggled to answer the question of how to protect sensor components from substances that naturally exist in extracted samples. A cross-disciplinary team of synthetic biologists at Northwestern University is developing a sensor platform that will be able to detect a range of environmental and biological targets in real-world samples. Using an established riboswitch to build a biosensor for fluoride, the team found they could both protect the sensor and operate similarly to the way cells do by encapsulating the sensor inside a fatty membrane. In a new paper published today, January 4, in the journal Science Advances, researchers demonstrated that by modifying the makeup and penetrability of the lipid bilayer membrane, they could further tune and control the performance of their sensor. So much data is being generated, and a lot of it is being driven by health apps like smartwatches, said Julius Lucks, a co-corresponding author and professor of chemical and biological engineering at Northwestern's McCormick School of Engineering. We can sense our heartbeat, our temperature, but if you think about it, we really have no way to sense chemical things. We're living in an information age, but the information we have is so minuscule. Chemical sensing opens enormous dimensions of information that you can tap into. Lucks is also the associate chair of the Chemical and Biological Engineering Department. His lab has advanced the field's understanding of molecular systems that respond to environmental changes by studying RNA and its role in cells, how RNA is used by cells to sense changes in their environment, and how these concepts can be used within cell-free systems to monitor the environment for cell-free synthetic biology, in which engineered biomolecular systems are used to activate biological machinery rather than living cells, is compelling because it is efficient, versatile and low cost. Lucks designed a riboswitch sensor using bacterial cell extracts to power gene expression reactions, including fluorescent RNA or protein that lights up in response to contaminants, that produce visual outputs cheaply and within minutes. Neha Kamat, an assistant professor of biomedical engineering within McCormick and a co-corresponding author, originally met Lucks at their faculty orientation and was interested in his desire to expand access to information. Kamat, whose expertise is in engineered membranes and membrane assembly, wondered if she could make Lucks's test tube system better using a vesicle, a membrane with 2 layers. They're using RNA and its associated machinery to sense molecules in real water samples and generate meaningful outputs, Kamat said. My lab works a lot with the lipids commonly used to encapsulate mRNA for drug delivery, with the goal of using these compartments to build more cell-like structures. We had the idea that we could protect Julius's switches and allow them to work in samples that might be kind of dirty with other contaminants, like a cell can. Other researchers have tried to place a sensor inside a membrane, but the switch stopped working properly and produced a much smaller sign. To overcome this, the team modified the genetic output in the sensor to amplify and color it, so it's visible by eye and, you don't need a fancy detector to do it, said Lucks. Encapsulation and protection are important to the sensor to make it function in native environments, like a wastewater channel with lots of other contaminants to erode the switch. This would be an example of distributed sensing, which could aid in fields from agriculture to human health. The group came together more officially when they received Northwestern's Chemistry of Life Processes Institute's, CLP, Cornu Innovation Award by pitching their potentially disruptive idea to the CLP's advisory board. The team earned seed funding to get their idea off the ground. Lucks calls this project a jumping-off point, from which they will be able to embed sensors into more materials, including smart materials that can change properties, as in biology. As synthetic biologists, one of our major themes is identifying challenges and looking to nature, Lucks said. What is it doing already? Can we build off that and make it do more to meet our needs? Fluoride became an obvious choice because there's a natural RNA molecule that senses it, allowing the team to design a simpler mechanism. But in the future, Kamat and Lucks have high ambitions about where use of the sensors can expand. For example, the sensors could flow through the human body to detect small molecules and biomarkers before the sensor is retrieved through urine or another passive method. It could also detect levels of nitrate in soil and aid in monitoring runoff. Beyond that, Lucks and Kamat are excited to see uses within materials science such as soft robotics, thinking about how to build something akin to a butterfly that smells through its feet. The paper, robust and tunable performance of a cell-free biosensor encapsulated in lipid vesicles, was supported by the CLP, the National Science Foundation, grant numbers 1,844,219, 1,844,336 and 2145050, and the U.S. Department of Defense National Science and Engineering Graduate Fellowship. Margreta A. Boyd and Walter Thavaraja, of Northwestern, were also co-authors on the study. Materials provided by Northwestern University. Original written by Wynne Reynolds. Note: content may be edited for style and length. Margreta A. Boyd, Walter Thavaraja, Julius B. Lucks, Neha P. Kamat. Robust and tunable performance of a cell-free biosensor encapsulated in lipid vesicles. Science Advances, 2023, 9, 1, doi, 10. 1126, Sci-Adv. Ad 6605 Northwestern University. Researchers detect fluoride in water with new simple color change test. Test is first to use artificial cell sensors to detect environmental contaminant. Science Daily. Science Daily, the 4th of January 2023. IFT. TT, BIC 721s. Northwestern University. 2023, January 4th. Researchers detect fluoride in water with new simple color change test. Test is first to use artificial cell sensors to detect environmental contaminant. Science Daily. Retrieved January 5, 2023 from ift.tt/bic721s Northwestern University. Researchers detect fluoride in water with new simple color change test. Test is first to use artificial cell sensors to detect environmental contaminant. Science Daily. https://ift TT, BIC 721s, accessed January 5, 2023. Thanks for watching. Please subscribe my channel for more videos.